The Crenarchaeota, Greek for spring old quality, as specimens were originally isolated from geothermally heated sulfuric springs in Italy, also known as Crenarchaea or eocytes. Archaea that have been classified as a phylum of the Archaea domain. Initially, the Crenarchaeota were thought to be sulfur-dependent extremophiles, but recent studies have identified characteristic Crenarchaeota environmental rRNA indicating the organisms may be the most abundant Archaea in the marine environment. Originally, they were separated from the other Archaea based on rRNA sequences. Other physiological features, such as lack of histones, have supported this division, although some Crenarchaea were found to have histones. Until recently, all cultured Crenarchaea had been thermophilic or hyperthermophilic organisms, some of which have the ability to grow at up to 113 degrees Celsius. These organisms stain gram-negative and are morphologically diverse, having rod, cocci, filamentous and oddly shaped cells. Sulfolobus. One of the best characterized members of the Crenarchaeota is Sulfolobus sulfataricus. This organism was originally isolated from geothermally heated sulfuric springs in Italy, and grows at 80 degrees Celsius and pH of 2 to 4. Since its initial characterization by Wolfram Zillig, a pioneer in thermophile and Archaean research, similar species in the same genus have been found around the world. Unlike the vast majority of cultured thermophiles, Sulfolobus grows aerobically and chemorganotrophically, gaining its energy from organic sources such as sugars. These factors allow a much easier growth under laboratory conditions than anaerobic organisms and have led to Sulfolobus becoming a model organism for the study of hyperthermophiles and a large group of diverse viruses that replicate within them. Marine species. Beginning in 1992, data were published that reported sequences of genes belonging to the Crenarchaea in marine environments. Since then, analysis of the abundant lipids from the membranes of Crenarchaea taken from the open ocean have been used to determine the concentration of these low-temperature Crenarchaea. See Text 86. Based on these measurements of their signature lipids, Crenarchaea are thought to be very abundant and one of the main contributors to the fixation of carbon. DNA sequences from Crenarchaea have also been found in soil and freshwater environments, suggesting that this phylum is ubiquitous to most environments. In 2005, evidence of the first cultured, low-temperature Crenarchaea was published, named Nitrosophomylus maritimus. It is an ammonia-oxidizing organism isolated from a marine aquarium tank and grown at 28 degrees Celsius. Eocyte hypothesis. The eocyte hypothesis proposed in the 1980s by James Lake suggests that eukaryotes evolved from the prokaryotic eocyte. One possible piece of evidence supporting a close relationship between Crenarchaea and eukaryotes is the presence of the homologue of the RNA polymerase subunit RBP8 in Crenarchaea, but not Euriarchaea. See also Euriarchaeota. Further reading. Scientific journals. Cavalier Smith. T. 2002. The Neoma and Origin of Archaebacteria. The negibacterial root of the universal tree and bacterial megaclassification. Int. J. Cyst. Evil. Microbial. 52. pt 1. 7-76. DOI. 10.1099/002077135217. PMID 11,837,318. Stackerbrandt. E. Fredrickson W. Garrity G. M. Greimann P. A. Kampfer P. Maiden M. C. Nesme X. Rosello Mora R. Swings J. Trooper H. G. Vorteran L. Ward A. C. Whitman W. B. 2002. Report of the Ad Hoc Committee for the Re-evaluation of the Species Definition in Bacteriology. Int. J. Cyst. Evil. Microbial. 52. pt3. 1043-1047. DOI. 10.1099 IJS. 0.023600. PMID 12,054,223. Gertler, V. May all B.C. 2001. Genomic Approaches to Typing, Taxonomy and Evolution of Bacterial Isolates. Int. J. Cyst. Evil. Microbial. 51. pt1. 3-16. PMID 11,211,268. Dalevi. D. Hugenholtz P. Blackwell Ul. 2001. A multiple outgroup approach to resolving division-level phylogenetic relationships using 16S rDNA data. Int. J. Cyst. Evil. Microbial. 51. pt2. 385-391. PMID 11,321,083. Keswani. J. Whitman W.B. 2001. Relationship of 16S rRNA sequence similarity to DNA hybridization in prokaryotes. Int. J. Cyst. Evil. Microbial. 51. pt2. 667-678. PMID 11,321,113. Young, J.M. 2001. Implications of alternative classifications in horizontal gene transfer for bacterial taxonomy. Int. J. Cyst. Evil. Microbial. 51. pt3. 945-953. DOI. 10.1099 00207713513945. PMID 11,411,719. Christensen, H. Bisgaard M. Fredrickson W. Mutters R. Kunit P. Olsinger. 2001. Is characterization of a single isolate sufficient for valid publication of a new genus or species? Proposal to modify Recommendation 30B of the Bacteriological Code. 1990 revision. Quote. Int. J. Cyst. Evil. Microbial. 51. pt6. 2221-2225. DOI. 10.1099 0020771351-6-2221. PMID 11,760,965. Christensen, H. Angen O. Mutters R. Olsinger. Bisgaard M. 2000. DNA hybridization determined in microwells using covalent attachment of DNA. Int. J. Cyst. Evil. Microbial. 53. 1095-1102. DOI. 10.1099 00207713503195. PMID 10,843,050. Zhu. HX. Kawamura Y. Li N. Zhao L. Li Trademark. Li ZY. Shu S. Ezeki T. 2000. A rapid method for determining the G+C content of bacterial chromosomes by monitoring fluorescence intensity during DNA denaturation in a capillary tube. Int. J. Cyst. Evil. Microbial. 54. 1463-1469. DOI. 10.1099 0020771350414663. PMID 10,939,651. Young, J.M. 2000. Suggestions for avoiding ongoing confusion from the Bacteriological Code. int j, cyst, evil, microbial, 50, 4, 1687 to 1689, DOI 10.1099/0020771350416887, PMID 10,939,677, Hansman, S. Martin W. 2000. Phylogeny of 33 ribosomal and 6 other proteins encoded in an ancient gene cluster that is conserved across prokaryotic genomes. Influence of excluding poorly alignable sites from analysis. Int j, cyst, evil, microbial, 50, 4, 1655 to 1663, DOI 10.1099/0020771350416555, PMID 10,939,673, Tyndall, B.J. 1999. Proposal to change the rule governing the designation of type strains deposited under culture collection numbers allocated for patent purposes. Int j, cyst, bacterial, 49, 3, 1317 to 1319, DOI 10.1099/0020771349313317, PMID 10,490,293, Tyndall, B.J. 1999. Proposal to change Rule 18a, Rule 18f and Rule 30 to limit the retroactive consequences of changes accepted by the ICSB. Int j, cyst, bacterial, 49, 3, 1321 to 1322, DOI 10.1099/0020771349313321, PMID 10,425,797, Tyndall, B.J. 1999. Misunderstanding the bacteriological code. Int j, cyst, bacterial, 49, 3, 1313 to 1316, DOI 10.1099/0020771349313313, PMID 10,425,796, Tyndall, B.J. 1999. Proposals to update and make changes to the bacteriological code. Int j, cyst, bacterial, 49, 3, 1309 to 1312, DOI 10.1099/0020771349313309, PMID 10,425,795, Berggraf, S. Huber H. Stetico, 1997. Reclassification of the Crenarchaeal orders and families in accordance with 16s rRNA sequence data. Int j, cyst, bacterial, 47, 3, 657 to 660, DOI 10.1099/00207713473657, PMID 9,226,896, Pallies, T. Nakamura L.K. Kohan F.M. 1997. Discovery and classification of ecological diversity in the bacterial world. The role of DNA sequence data. Int j, cyst, bacterial, 47, 4, 1145 to 1156, DOI 10.1099/0020771347411145, PMID 9,336,922, Userby, J.P. 1997. List of bacterial names withstanding in nomenclature. A folder available on the Internet. Int j, cyst, bacterial, 47. 2. 590 to 592. DOI 10.1099 00207713472590. PMID 9103655. Clayton, Ra. Sutton G. Hinkle P.S. Jr. Bolt C. Fields C. 1995. Intraspecific variation in small subunit rRNA sequences in GenBank. Why single sequences may not adequately represent prokaryotic taxa. Int. J. Cyst. Bacterial. 45. 3. 595 to 599. DOI 10.1099 00207713453595. PMID 8590690. Murray, R.G. Schleifer K.H. 1994. Taxonomic Notes. A proposal for recording the properties of putative taxa of prokaryotes. Int. J. Cyst. Bacterial. 44. 1. 174 to 176. DOI 10.1099 00207713441174. PMID 8123559. Winker, S. Wozer C.R. 1991. A definition of the domains Archaea, bacteria and eukarya in terms of small subunit ribosomal RNA characteristics. Cyst. APPL. Microbial. 14. 4. 305 to 310. DOI 10.1016 per second 0723-2020. 11. 80303-6. PMID 11540071. Wozer, C.R. Candler O. Wheelis M.L. 1990. Towards the Natural System of Organisms. Proposal for the domains Archaea, bacteria, and eukarya. Proc. National. ACAD. Sci. USA. 87. 12. 4576-4579. Bibcode 1990 PNAS 87.4576W. DOI 10.1073 PNAS 87.12.4576. PMC 54159. PMID 2112744. Achenbach Richter, L. Wozer C.R. 1988. The ribosomal gene spacer region and Archaebacteria. Cyst. APPL. Microbial. 10. 211 to 214. DOI 10.1016 per second 0723-2020. 88. 80002X. PMID 11542149. McGill, T.J. Jerka J. Sobieski J.M. Pickett M.H. Wozer C.R. Fox G.E. 1986. Characteristic archaebacterial 16S rRNA oligonucleotides. Cyst. APPL. Microbial. 7. 2 to 3. 194 to 197. DOI 10.1016 S0723-2020. 86. 80005-4. PMID 11542064. Wozer, C.R. Gupta R. Hans C.M. Zillag W. 2J. 1984. The phylogenetic relationships of 3 sulfur-dependent archaebacteria. Cyst. APPL. Microbial. 5. 97 to 105. DOI 10.1016 S0723-2020. 84. 80,054-5. PMID 11,541,975. Wozer, C.R. Olson G.J. 1984. The phylogenetic relationships of three sulfur-dependent archaebacteria. Cyst. APPL. Microbial. 5. 97-105. DOI. 10.1016, S0723-2020. 84. 80,054-5. PMID 11,541,975. Wozer, C.R. Fox G.E. 1977. Phylogenetic structure of the prokaryotic domain. The Primary Kingdoms. Proc. National. ACAD. Sci. USA. 74. 11. 5088-5090. Bibcode. 1977 PNAS. 74.5088W. DOI. 10.1073 PNAS. 74.11.5088. PMC 432104. PMID 270,744. Scientific books. Garrity G.M. Holt J.G. 2001. Phylum AI. Crenarchaeota Phi. November. Quote. In Dr. Boone. R.W. Caston-Holtz. Bergey's Manual of Systematic Bacteriology Volume 1. The Archaea and the Deeply Branching and Phototrophic Bacteria. 2nd ed. New York. Springer Verlag. p. 169. ISBN 978-0-387-98771-2. Scientific databases. PubMed references for Crenarchaeota. PubMed Central references for Crenarchaeota. Google Scholar references for Crenarchaeota. NCBI taxonomy page for Crenarchaeota. Search Tree of Life taxonomy pages for Crenarchaeota. Search Species 2000 page for Crenarchaeota. MicrobiWiki page for Crenarchaeota. LPSN page for Crenarchaeota. Crenarchaeota from the University of Wisconsin Virtual Microbiology Site. Comparative analysis of crenarchaeal genomes. At DOSE IMG System.